second half of the mark scheme. Let's go through this nice and fast. Which one is not a loaded coenzyme? So anything that doesn't have a plus on it is really easy to go with. And ATP is a loaded coenzyme, so is NADPH, and so is NADH. Going on here, a student was asked to identify the overall process of photosynthesis and respiration in eukaryotic cells. They prepared the following table to outline the differences. So, photosynthesis, exergonic. Does it produce energy or does it need energy? No, so it's definitely wrong there. All stages occur within chloroplasts. Yes, all stages occur within mitochondria. No, because glycolysis is there. Electron transport not involved. We had C. No, there is electron transport involved in photosynthesis. It's a bit of the old study design there. Uses water as a react. This has got to do with the photosystem. Sorry, going back. Uses water as a reactant in the first stage. Yes. Forms water as a product in the final stage. Yes. So even if you weren't sure about the electron transport bit, you could probably get there based on that last one. And this is a pretty typical exam question. They won't ask you really obvious ones. They'll ask you to pull together a whole bunch of information in one multiple choice question. So how are cellular respiration and photosynthesis almost opposite? Essentially, photosynthesis is going to remove the carbon dioxide and cellular respiration is going to put it back. Photosynthesis releases energy. No, it doesn't. Photosynthesis releases oxygen from the atmosphere. No, it removes oxygen. Sorry, no, it doesn't. It removes carbon dioxide, or all of the above. And because they're wrong, that one can't be correct. All right, let's have a look here. This is my favorite question of all time. And it's really hard. So this one is asking you, what is occurring at point M? So the first question asks you, what is a point M? So this is the rate of carbon dioxide usage. Yeah, so rate of carbon dioxide exchange between its leaf and its environment as light intensity is altered. So what's happening down here is the light intensity is really low. So that means that photosynthesis is happening, but it's just happening really, really slowly. And as it happens really, really slowly, you can see that carbon dioxide starts to be used up. But why is it negative? Why is there negative carbon dioxide used when there's not much sunlight? I want you to think about the golden rule of cellular respiration. So long as you are respiring, you are alive. So plants are alive, they're respiring. So what's happening down here is that the rate of cellular respiration is actually higher than the rate of photosynthesis. And up here, the rate of photosynthesis is higher than the rate of cellular respiration. So where they equal out, where they are completely normal and completely on the same level, is that you've got respiration, releasing the carbon dioxide. You've got photosynthesis, taking the carbon dioxide. Bang. That's where you end up with the rate of photosynthesis is equal to the rate of cellular respiration. So what's happening above 600? Essentially what I was looking for here was anything else limiting. And I would like you to mention what is a limiting factor. The last one, why is it harmful for a plant to have a low level of carbon dioxide? You've got less carbon dioxide, which means less glucose produced, and that's 1 point. Yeah, 1 point for the glucose point. Then the next one was if you get more ammonia or hydrogen peroxide, it's toxic to the plant. So usually we had people giving me one or the other, but not both. Now the last one was our experimental design question. So I'm not going to read through the STEM, but why were the isolated mitochondria used versus the whole cells? If you isolate the mitochondria, you remove the influence of glycolysis. Glycolysis produces ATP, and so therefore you can't tell if you leave the cytosol in whether or not the result is due to mitochondria or it's due to glycolysis. Now, the next one down here, again, lots of people gave me really generalized answers. It's important to know that if you are given an input, you need to tell me where it's inputting and where is the output. So why can the uptake of O2 be used? Let's just focus on the uptake of that. And that's because oxygen is used or an input or it's a reactant in the electron transport chain. And I needed that link, not just used in cellular respiration. That is too general. You must tell me where it's used in cellular respiration. Same goes with carbon dioxide. So carbon dioxide is a output or a product of the Krebs cycle. If you didn't tell me it was Krebs, then I couldn't give you the mark. Let's get this done. Why must all factors except the glucose and pyruvate be kept the same? A lot of people talking about accuracy and reliability. Reliability has to do with repeating the experiment. Accuracy has to be how close to the true value. What we're doing by keeping things the same is that we are having one independent variable, and one independent variable refers to validity. So can you make sure that you kind of practice it in your brain and you go, yep, 1 IV, that means I'm valid. Now here we had a 2-mark question. Why were the mitochondria given glucose so inactive while those given pyruvate so very active? So 2 parts, 2 dot points, split your question. You could even split it with a table. And let's go here. So the first one, mitochondria can't use glucose. They can't use them, and there's no cytosol to do glycolysis. So they can't use it, and they're not going to produce ATP. The second one, the pyruvate can be used by the mitochondria for the Krebs cycle. So therefore, yay, we're going to get lots of activity. And that's it. Split it in half. Do one bit on either side of the question. You can make it a table. It doesn't matter. So the last question on this page, why did the mitochondria given glucose show a small amount of activity at the beginning and then stop? Okay, and that's because the cells themselves will have already contained some pyruvate. Okay, so they had some glucose, some pyruvate, use it up, and then run out. So a lot of people could tell me that it had run out, and that's why it stopped, but forgot to tell me that they had some pyruvate at the beginning. Now, the very last question, pretty well answered as far as we go. This one here, a lot of people thought we were talking about, because it said seeds, you went to photosynthesis. No, the key here is that they're respiring. So this whole question is about respiration. So what is the gas produced during respiration? Carbon dioxide. Why did the water level rise less during these hours? Because the oxygen is used up really fast, and then there's less oxygen available, so it's used up slower, which means less CO2 is being produced. And so there are your 2 points. The oxygen is being used up at a slower rate, and the carbon dioxide is therefore being produced at a slower rate, so your water level doesn't rise as fast. And the last one, if you didn't get part 1, it's very hard to get part 3. The inputs that produce carbon dioxide, we get NAD+, ADP plus PI, if you mentioned it. I don't need numbers here, but if you wanted to put 2 there, that's fine. And the last one is pyruvate is also needed. And that's the end of your test.